to plug it in. It's not even plugged in here.
No.
That should be the switch, yes, if it works.
Okay, so
Okay, welcome back to RegSys. My name is Julia Seidlinger, and I'm going to chair the last session for today. We're going to continue on regulatory elements, and the first speaker, another keynote speaker, is Robin Andersson, and he's going to tell us about his results on using cage data, basically, and comparing enhancers and promoters. So, please.
All right, so thank you very much for inviting me to present our work here. So, as most of you, I'm very fascinated by how a single genome can drive such a large diversity of cell types with sometimes very different functions and morphology, as illustrated here by neurons and pancreatic islet cells. So my group is interested in how these diversities are regulated and how this regulation is encoded in the genome. So we take a transcription-centric approach and to model regulatory elements, their activities. So we're interested in promoter and enhancer function. So to get all on the same page to start with, we consider promoter function the ability to recruit RNA polymerase II and initiate transcription locally. We consider enhancer function the ability to enhance recruitment and/or transcription at distal locations. Right. So, in my talk, it's divided into two parts. I would start by describing our efforts to localize regulatory elements based on transcription initiation data and how we can use that kind of data to determine the activity, the enhancer and promoter activity of regulatory elements. And that will also lead to our current thoughts about the need to perhaps revise the definition of a regulatory element. More on that later. In the second part, I will talk about our recent efforts to use expression data to model regulatory topologies and regulatory redundancies and how we can make use of this to say something about the importance of individual elements. So to model transcription regulation, as I said, we take a transcription or expression-centric approach. So, for instance, consider this particular RNA as what people mostly do with RNA, if you want to measure their abundance in cells, is that you use RNA-seq, and all of you know that. So basically, you chop up the RNA, you sequence those, and often you map these to a reference genome if you're taking a second-generation sequencing approach. Using that technique, we can, of course, then estimate the transcripts, the splicing that happens, but also the abundance of RNA. So the technique that we are mainly using is called cap analysis of gene expression, or CAGE, which is not considering the whole RNA, but the 5' end of capped RNAs. And if we map these to a reference genome, so we get peaks like this, that will indicate the location of transcription start site, and we can use that to then identify core promoter regions. And similar to RNA-seq, the amount of cage reach we get gives you an abundance, a measure of the abundance of the RNAs in the cell. So if you're interested in cage data, in the analysis of cage data on other 5' RNA sequencing data, there's a nice new R package out called CageFighter, developed by Malti in Albin Sandelin's group. So this should work on other kind of 5' end sequencing data as well. So this is how cage data look like, not just the cartoon version of it. So here is a RegSys gene with the annotated transcription start site here, and you see a peak of the location of the 5' end of the reads indicating that this is the transcription start site. So this is an assay that was used in the Phantom project, and in the Phantom 5 project we had a quite ambitious aim that was to map all transcription start sites across human and mouse, across the majority of cell types and tissues. So some interesting findings that came out of this is that this is the annotated TSS. The most preferred TSS of this particular gene is actually 200 base per downstream in the 5' UTR. And if you look across different cell types, you can see that the preferred transcription start site across cell types differ. So this is, for instance, it's showing you aortic smooth muscle cells. Here you have T cells, etc. So, and the third finding that I wanted to highlight is the sheer amount of transcription start sites that were identified. And regardless if we were using a lenient or a strict thresholding on the expression level of these, the majority of transcription start sites identified were not associated with any mRNA gene promoters. So there seems to be a pervasive transcription initiation happening in the genome. But most of these RNAs are likely not functional or interesting, and many of these are actually degraded by various pathways in the cell. And one of these is the exosome complex that I will talk a bit about. So here I show you another example. This is in HeLa cells, and in this time you can actually see that the cage-inferred transcription start site is very similar. It agrees well with the annotated transcription start site. So what we did here in collaboration with Torben Haik Jensen is to knock down the exosome, and then we did perform cage before and after this exosome knockdown. And if you do that, you can see a new transcription start site coming up just upstream of the mRNA transcription start site in the antisense direction. And these are called prompts for promoter upstream transcripts, or UARNAs, upstream antisense RNAs. And they were discovered by 3 independent labs, Torben Jensen labs, Phil Sharp, and John Liss labs, basically at the same time. So what we see here is something that we refer to divergent transcription. So if we now look genome-wide and at these transcription initiation sites and split these according to their genomic annotations, we can see some interesting stuff happening. So first we can see that, so this is now on that will correspond to the mRNA. For protein coding gene, you have a look at the blue. Most of mRNAs are very stable, not degraded by the exosome. If you have a look at link RNAs, you can see that the majority of these are actually degraded, right? And now having a look at the prompts, those would be the one originating and going in this way. Most of these are actually very unstable. So if we now use cage data to look at human gene promoters and say, how frequent is this divergent transcription? So just requiring only one read tells you that almost all of them are divergently transcribed. But we know that prompts are low abundant, they are degraded and so on. So if we impose strict expression thresholds, we will of course see a lower fraction, right? So what we did here was to sample expression levels from genomic background regions likely not containing any transcription start site. So that was like informed sampling using DNase regions, etc. We can get some kind of noise threshold and estimate that as many as 82% of gene promoters seem to be divergently transcribed. And we can do the same using nascent RNA sequencing data. This is encapsulated GrowCap in different cell lines, and we get quite similar estimates. A huge majority of human gene promoters are actually divergently transcribed. So now, is this noise, or is there any kind of information directing transcription initiation at these sites? So if we do motif searches for known core promoter element, like the initiator or the Tata box, we can see that the sequence information directing transcription start site and the direction of transcription is actually quite similar at prompts as for mRNAs. We can also map SHIP exodata from the Frank Pugh lab and see that there are general transcription factors actually binding to these core promoter elements. So there is sequence information directing the transcription and the transcription initiation at these unstable RNAs. So our view of gene promoters has changed quite a lot the last decade or so, and we now know that gene promoters are characterized by open chromatin. They are characterized by a pair of divergent core promoters that initiate divergent transcription, and this often happens just at the boundaries of flanking nucleosomes. And also importantly, gene promoters are just not arbitrarily defined by a certain, let's say, let's call them 1 kb long, etc. But often we see that transcription factors are actually binding within this open chromatin region. So this gives, in my opinion, a quite elegant description of what a gene promoter is. All right, so we can use cage data to infer transcription start site, to map core promoter elements, and that tells us quite a lot about gene promoters, but in order to model transcription in regulation, we also need to know about the activity of enhancers. So for what I said, my transcription-centric view, a quite interesting observation now almost 10 years ago, came from 2 papers that show that when you stimulated cells, so this is in mouse macrophages and this is in mouse neurons, when you stimulated these cells, enhancers were bound by RNA polymerase. And this is something that we have known for a while at individual enhancers, but now they saw a massive effect. And with RNA-seq data, they could show that enhancers were divergent to transcribe when activated. So we know today that both gene promoters and enhancers are divergent to transcribed, but they have different transcriptional signatures. So as I showed you a couple of slides back, mRNA gene promoters often more biased to the sense direction, the mRNA direction, whilst the prompt transcription is a bit, of course, lower. So this signature is quite interesting, but it's also useful because we can use this signature to predict the location of enhancers genome-wide. And this is something that we did in the Phantom project, where we used cage data alone to, what I would say, create the most extensive atlases of transcribed enhancers across human and mouse cell types. And this was done in a lot of different cell types, but also across a lot of different time courses that is stimulation-based and differentiation-based. So if you're interested in using this data, here are the sites where you can download the predicted location of enhancers and the activity across cell types. And when we published this was in HE19 and MM9. We have now updated the data, so we have it in MM10 and HE38 as well, and you can download this from these Zenodo repositories. So, is enhancer transcription, is that noise? Is this just random transcription initiation events happening in the genome? Well, similar to what we did at prompts, we can have a look at the sequence content around transcription start site at enhancers. And we can actually see that there are core promoters, sequences that resemble core promoter elements like TOTA boxes and initiator at the suspected location around enhancer transcription start sites. We can also have a look at the location of transcription start site with respect to the chromatin, the local chromatin. And if we do a cross-correlation of the position of the 5' end of a caged read versus the 5' end of an MNase-seq read that will tell you the location of nucleosomes, we can see that the most preferred lag is actually just around 0. So suggesting that the transcription initiation at enhancer is also just happening just at the boundary of flanking nucleosomes. All right, so this was in human and mouse. So Sarah and Maria in my lab, they boldly decided to go against the Drosophila geneticist crowd that had an opinion, at least, I know that several had, I'm not saying that all of you having that the Drosophila genome is a bit more pure, we don't have prompt transcription, we don't have an enhancer RNase, etc. This is something weird happening with human and mouse. So what they decided to do was to do a very similar approach that we did in HeLa cells. So again, together with Torben, we did a knockdown of the exosome. This time we considered the catalytic component RP6 and DIS3, and then we did cage before and after this knockdown. So here you can see now this time Drosophila gene transcripts, And this time you can see different transcription initiation sites for these particular transcripts. These are 4 replicates, so you can see how nice the data actually is, right? If we now look at the exosome knockdown data, you can see that prompt transcription is showing up here, right? The red one is going in this direction, in the minus strand direction. So we also had a look at enhancers. So this is now a star-seq predicted enhancer from Alex Stark lab. In the wild-type cage data, we basically don't see any transcription initiation. If we knock down the exosome, we can see this coming up, suggesting that it's actually divergent transcription happening at Drosophila enhancers as well. So we estimate that in flies, at more than 50% of standalone mRNA promoters are divergently transcribed, and more than 80% of transcribed enhancers are divergently transcribed as well. But this is, of course, dependent on the assay you use, the threshold you used, etc. But basically, I think this is interesting that we see such a strong resemblance between so distal species. All right, so to sum up that part, our current view of enhancers is that they are defined by open chromatin. They are bound by transcription factors. They are characterized by a pair of divergent core promoters that initiate divergent transcription just at the boundary of flanking nucleosomes. And you might recognize this figure, and this is because it's exactly the same I showed you a couple of slides back. And the reason why I did that was that mine, and I know many others now, believe that gene promoters and enhancers are actually very similar in their architectures. But there are some differences, so I wouldn't suggest that they are exactly the same. And the main differences that we can see is the amount of RNA produced from these, right? We know that mRNAs are more abundant than the enhancer RNAs. They are degraded differently, etc. And this is mainly driven by DNA elements that are flanking the enhancers. So if you have an excess of polyadylation sites and a low number of 5' splice sites, you tend to terminate transcription early, which leads to non-polyanylated transcripts that are degraded by the exosome. So you might argue that there are differences between gene promoters and enhancers in the level of histone 3 lysine 4 monomethylation and histone 3 lysine 4 trimethylation. Well, this is true in many cases. So, but if we now compare open chromatin regions, transcribed open chromatin regions, according to the level of transcription, you can see that histone 3 lysine 4 monomethylation is associated with low transcription, and histone 3 lysine 4 trimethylation increases with increasing transcription levels. So if you're using the ratio between monomethylation to trimethylation to predict enhancers, you're basically selecting for regions with low promoter activity. And that might be convenient, but you might miss out on some really interesting biology here. So we can look at this in a bit more detail. So what we did here was to split open chromatin regions into those overlapping mRNA gene promoters and those that are distal from mRNA gene promoters. So these are potential candidate enhancers in this case. And what we did next was to split these according to the transcription level, this time by GrowSeq. And most candidate enhancers actually end up in these 2 categories with low transcription, and most gene promoters that are transcribed end up in these categories. So what you can definitely see here is that we do have more histone 3 lysine 4 monomethylation at candidate enhancers, and we do have more histone 3 lysine 4 trimethylation at gene promoters. But if we compare across transcription, if we pair up the transcription, you can actually see that these histone modifications are very similar between these categories. So I am not arguing that there is any causal or anything driving any causal relationship between the transcription level and histone modification. And I know that this is being debated. There was a preprint out just 3 days ago from Jane Mellor's lab that could not see any relationship at all. So this is still ongoing work. But so some interesting things that we found in the Phantom project when we wanted to validate our prediction is that transcribed enhancers tended to validate much better than untranscribed enhancers in vitro. So that suggests that regulatory elements with enhancer potential that also regulatory elements that have some form of promoter activity tend to have enhancer potential, which is now starting to get a bit, we can also see that transcription informs on cell type specific activity. So one of the things that we did was to select certain enhancers based on their expression profile in human. So this is one enhancer, which is specific to neurons and also a bit of expression in astrocytes. We then did a reporter construct and did a stable integration with these in zebrafish embryos. And you can see that these are primarily used in the brain in the neural tube in zebrafish. We suggest that the expression tells you something about their cell type specific activity as well. I will skip this because I'm running out a bit of time. So what I think we have here, it's not yet, right? Now, all right. So to conclude this first part of my talk, I strongly believe that what we have is a unified architecture of regulatory elements. We know that enhancers and promoters are very similar, and there's actually some problems here with terminology. So I strongly believe that we need to revise this dichotomized view of enhancers and promoters as very distinct elements. Maybe we should consider them regulatory elements that could have different functions instead, but these functions are definitely not mutually exclusive. There are several reports now showing standard, very classical mRNA gene promoters having enhancer activity. We know that many enhancers have promoter activity. This is how we identify them with cage data. All right. So we can use expression data to identify regulatory elements, their activity, and can we now use this to tell something about the topology of transcription regulation? So Sarah in my group had this idea that maybe we could split expression into two major components. One, so we will be able to derive the chromatin topology from expression data itself. So the idea here is to think about expression as a result, or transcription as a result of many features. And one of these features is guiding or telling, one of these features is the component of how much expression is explained by its genomic position in the genome. So she used approximate Bayesian modeling in which she had this positional dependent component modeled by a random walk, in which we tried to minimize the differences between consecutive genomic regions. So what does this kind of modeling give you? So here we have 3 different cell lines. This is showing the positional independent component. The other things that are not explained by position, and this is the position dependent component, things that we can explain by the genomic position of a regulatory element. So what you can see here is that these 3 cell lines are actually very similar in the profiles, and you have these kind of broad profiles. But if you zoom in, you can see that there are some differences. I highlight some here. And if we zoom in on this particular region over here, you can start to see differences between cell types. So in this particular one, we see this NOS1AP gene, which seemed to be repressed in GM12878 cell line. You see a negative positional component, and you see repression by K27 trimethylation, whereas the other cell lines have K36 trimethylation. So in general, what we can see is that this positional component of expression is, when positive, is reflecting active chromatin, and when negative is reflecting the B compartment, or the constitutive or facultative heterochromatin. And this is perhaps easy to see just at the compartment boundaries where you see this shift in signal. So if you're interested in the transcriptional decomposition approach and how we can use this to predict differences in chromatin architecture between individuals, you should have a look at Lucas' poster tonight at number E43. All right, so given now that we can use expression data to identify chromatin compartments and also expression domains, topological domains, I didn't show you that part, we had an idea that maybe we can use this to predict interactions between regulatory elements. So using a quite simple approach, random forest modeling, where we had positives defined by promoter capture high C interactions, Sarah predicted enhancer promoter interaction. And you can see that the AUC is actually quite nice. We still can improve on the precision recall, but I think this is really nice that we can use expression data alone just to infer the interactions between enhancers and promoters. So based on the model defined in GM12878, Sarah then went back to the phantom data again and took 76 cell types for which there are no high-C data available at the time when she did the modeling. And then did the transcription decomposition and the enhancer-promoter interaction predictions. So what you can see is that for most cell types, there are actually very little shared interaction between cell types, but similar cell types are clustering together here. So this is now a resource that we can use to try to understand transcription regulation, but I would also like to highlight how we can make use of it. So we had a look at GWAS-associated, disease-associated SNPs from GWAS, and in particular we could see that monocytes were enriched in Crohn's disease for disease-associated SNP interacting with promoters. So in this example here we have this is the prostaglandin receptor 4, where you can see this is predicted interacting with GWAS-associated SNP in a cluster here, 200 kb upstream of the gene, where we have many predicted enhancers. All right, so in order to understand the impact of genetic variants, we then need to understand the topology of transcription regulation. But we also need to understand the importance of individual regulatory elements. And in particular, something I find very interesting that we're starting to look at now in my lab is the robustness of transcription regulation, or the potential redundancy between elements. So Scholli, a PhD student in my lab, he started looking at so he revisited the cage data from Eileen Furlong lab, which is was made on 81 inbred fly lines from the GGRP and across 3 different developmental time points. So the main questions that he was interested in here was how variable are core promoters in their activities, and how much of this variability can be explained by genetic variants. So the strategy here was to do a careful decomposition of the cage data into core promoters, and then do an EQTL, promoter EQTL analysis on top of that. So what he found out with this, he took a gene-centric approach after that, and what he found out was that the majority of cases, over 5,000 genes, have EQTL associated with a single promoter of that gene. So even if you have multiple promoters of the same gene, only one promoter was affected by association. It then also found more than 1,000 genes where there were multiple promoters of the same gene associated with the same EQTL. And many of these were showing a unidirectional behavior. So this is in this case now, where we have two promoters, and for instance here, the variant, in the variant allele, we see an upregulation of both promoters. But interestingly, what I found most interesting in this case is what we termed switch EQTLs, and where we had at least two promoters showing the opposite effect of the variant. So this is promoter 1 going down in the alternative allele, and promoter 2 going up in expression in the alternative. All right, so this is showing you the cage data for this particular example instead of the box plots. And what I find most interesting with this is that if you look at the gene expression level, you can see that this switch is actually, in many cases, rescuing the total gene expression level. So while this particular example here is showing a switch which is very proximal, maybe you should say that this is the same promoter. We have cases where there's a separation between these core promoter switches for several hundreds of base pairs. So what kind of genes are these? We can see that compared to other EQTL-associated genes, they tend to be more highly transcribed. They tend to be less variable across development, suggesting that these are more housekeeping-like genes. There's also an enrichment of certain core promoter elements like motif 1, DRE, and EBOX that also well characterized in Drosophila as housekeeping core promoter elements. So I should say now that the results here are related to what was already published by Eileen Furlong's lab previously when they identified these shape EQTLs. Well, what I think we have here is that we identified a general phenomena in which alternative core promoter elements that sometimes can be quite distal from each other they confer some kind of regulatory robustness to housekeeping genes. So we're not only working on Drosophila, as I told you, we have a big project running in the lab where we are looking at individual variation across lymphoblastoid cell lines. So we've done cage on more than 100 lymphoblastoid cell lines that were genotyped in the HapMap and the 1000 Genome projects. And these have been done also with certain stimuli. So Shirley and Nicholas are the main drivers in this project, worked quite a lot on this data. So if you're interested in how sequence is directing transcription in lymphoblastoid cell lines and how we can infer that kind of information from deep learning, you should have a talk with Nicholas at his poster E56 tonight. So what Shirley did using this data was to follow up on Drosophila results. And interestingly, he found many cases of this kind of robustness also happening for human genes. So this seemed to be a conserved robustness both in human and fly. I should say that it's much easier to analyze the Drosophila data because these are basically haploid, the inbred, and we don't have these massive problems with LD blocks in the same way in human. So it has been a bit more challenging, this part. All right, so to summarize, I've described what I think is a unified architecture of transcription regulatory elements, and this is something that has been observed in human, mouse, fly, to some extent in C. elegans and yeast. So I think this is a universal property of transcription regulatory elements. I've talked about how we can use expression to inform on regulatory activity and larger regulatory topologies using transcription decomposition. And I've explained how we can use individual variation data to study robustness and redundancy between regulatory elements. So I would like to end by thanking you for your attention, of course, and acknowledge the people that have been involved in this work. So I mostly presented work by Maria, Sholly, and Sarah, and this has also been some very nice collaboration with Torben Haik Jensen Lab, Albin's lab, and Piero and Yoshihide, part of the Phantom Consortium. Thank you.
Hi, Robin. Great talk. I was wondering about this switchy QTL. So is this the same variant that actually regulates two promoters, or could it be that these are two variants that are just very close to each other and they would act independently of each other?
Yeah, so in this case, it's the same variant that is associated with the two promoters. We cannot say anything about causality, of course, now, but and you can have in Drosophila. What I said, the nice thing with this is that basically the LD drops extremely quickly. So sometimes you don't really need to think about what is a lead SNP and what are proxy SNPs and so on. But of course there can be multiple SNPs showing the same association. Yeah.
Okay, and then it could be one for one promoter and one for the other promoter, essentially.
Yeah, but we also have many cases where we can only identify one.
Okay.
So
Okay,
yeah.
cool.
Yeah.
Thanks.
Great work. So this new concept of enhancers and promoters being in a spectrum rather than being discrete regulatory elements, I'm aware of at least multiple different people like John Liss, Victor Corses, I think Mikhail Spivakov has recently something. And from our data in human immune cells, what we are seeing is that a portion of these promoters are interacting with other promoters, even though they are not active themselves. They are acting as enhancers, driving the expression of other promoters in a genotype-dependent manner. So are you able to look at the same events, these three-dimensional interactions in the fly, and see whether these promoters can be acting as enhancers?
So all of this is quite tricky, and I think we understand gene distal enhancers much better because this is where the focus have been. So what I think is happening now, and what can tell us a lot about this, is this current trend of all these massively parallel reporter assays, showing that many gene promoters have enhancer potential. Then understanding if that is actually working in vivo can be tricky, right? Mm-hmm. Because just removing that promoter could have a lot of other effects, right? So this is the challenge that we have. But I think what I think we need to do is to try to not fall into this dichotomy of enhancers and promoters as such a difference, but be a bit more open-minded, because I think then we can start to rediscover these kind of things and the mechanism driving this, the enhancer potential of gene promoters, for instance.
Thanks.
Yeah.
Great talk, thank you. Can you comment on these predicted enhancer promoter interactions? How do they differ from the ones that you had in your Nature 2014 paper based on just correlation of the cage promoter and enhancers? Because I think those are still very widely used by open targets, for example, to map GWAS SNPs to their target genes. So
what we did there was a very simple approach where we just looked at correlation, basically with the assumption that if the enhancer is active in one cell type, and you also have a promoter active in the same cell type, and then you look across many cell types, and if you have some kind of co-expression, we could associate them, right? The problem with this is that, of course, we have the low enhancer transcriptional levels. That's one problem. And also that there might not be a strict linear relationship in this. So what this is allowing us to do now is, for one, consider correlation, but also take into account other properties that we can derive from the decomposition, as when is the positional component dropping, right? Which is in many cases tell us the boundaries of these topological domains, for instance, and often have this kind of information is now also considered in this model.
So this should be essentially, I guess, a subset which is less prone to having false positives in there.
Yeah. But let's discuss it.
I want to follow up on Judith's question about the switch promoters, actually. So you mentioned that sometimes they're actually quite far apart from each other. So what does far here mean? Like, they still in the same accessible chromatin region, or would you even see effects to alternative promoters that are in separate nucleosome-depleted regions? Because that then I would be interested in how you would explain that, what your ideas behind that are.
So definitely they are separated. They can be separated very proximal, but it can also be separated at distances that cannot fit within the same promoter as defined by the open chromatin. So what I think is happening, we don't know the mechanism. We know that there are other experimental data, like from the Asifa Akhtar lab on this, on the NSL, on the MOF, seeing some of these properties also as housekeeping genes. So what could happen is that if that you have one preferred promoter, but you also have a redundancy in the sequence that will allow some other regions to take over. If that is, there might be some kind of competition, or we don't really know that yet. Yeah.
So. Of course, you keep saying there's a continuum between enhancers and promoters, but we also know there are widely different promoter types as well.
Yeah.
And I'm not sure how those fit on that continuum. For example, you showed that your bidirectional promoter signal with prompts also has some data signal in them. Have you found any prompts that have actual canonical data that it is used to drive them? I couldn't find any.
You mean looking at individual elements and looking at the consensus sequence? This is what you mean? Because basically
I'm just saying that Tata and DP in Drosophila are intrinsically directional elements.
Yeah.
So to order to have prompts in another direction, you have to have another Tata or another DP in another direction. You can't have it from one, right?
Yeah.
So how does it look in those cases?
So we haven't in that way characterized we have looked at it a bit, right? I don't remember exactly now, but if we do motif matches, or if we look at, or if we extract the from the sequence the de novo calling, we see things that resemble these at least, right? Then how well they separate between different promoter classes, I cannot really tell you right now.
Yeah. So and for the switch promoters, we actually now pretty sure that for broad promoters, including the housekeeping promoters in Drosophila, the actual distribution of TSS peaks is determined by a stable position of +1 nucleosome. Have you actually looked at whether this change in the shift
that we haven't. That's a good idea. We haven't looked at that yet. Yeah, no.
Position. Okay, thanks.
Oh, since we're still getting ready, so one more question.
Okay, thank you. So there are several machine learning methods based on, for example, histone-modification chip-seq data, which aim to predict enhancers. Are those completely useless? Should we only measure this nascent RNA?
Well, so how do you validate these? So one of the big problems in the field is that we don't really have any good gold set of the true enhancers that we know are active in the particular cell types you're looking at, right? So often what we do is that we make some predictions of enhancers based on some assumption, like they are gene distal, for instance, or they have P300 or CBP or something like that, and then we find these histone modifications, and then we make models based on that, and then we predict, and then we overlap with some other predictions, right? So it becomes circular, and it becomes very, very strong ascertainment bias in what we are actually predicting.
Yes, I have noticed this.
Yeah. So this is a problem, and I think we need to have a combination of a lot of different. I should wrap up, but let's discuss this. Sorry. Yeah.